Hey guys, I wanted to talk to you a little bit about blood typing. I know this is kind of an odd way to do this, but it's best if I can show you, kind of in layman's terms, how I would explain blood typing to a student. Because in my mind, when you write out how this works, it makes it very easy for you and you won't miss any questions. And so I want to go ahead and illustrate that for you guys today. So I've got the 4 different types of blood types, and what I'm going to do is draw different cells and describe for you the antigen and antibodies that each one is going to exhibit. So we'll look at type A blood, we'll look at type B blood, AB blood, and O blood. So those are my red blood cells. And I'm going to use 2 different colors here to illustrate antigen versus antibody. So an antigen, remember, is going to be our surface marker. And I'm just going to write that at the top. And these guys, in my mind, this antigen is going to determine what blood type you have. Okay, so I always tell students, if you can kind of get in your head, the big picture, this shouldn't be too difficult. I know it is confusing whenever you're trying to do these questions. And so I'm going to draw our antigens in red. So remember, if I'm type A, that means my antigen, because that determines what blood type I have, has to be A on those. So I draw little A's on the surface of these cells. So an antigen, again, is a surface marker on a cell. In this case, I'm type A, so I have to have A antigens on the surface of my cell. I'm gonna not go on and do these. I'm gonna continue with type A first and talk about antibodies, and then we'll come back and we'll do those together as well. When you look at these type A situations, what you have here is, because I have an antigen A, I also have to have an antibody. So in this case, if my whole blood was all together, okay, so we'll put that cell in a beaker. My blood would also then contain an antibody. An antibody protects your antigens. So if you have the same antigen that you have antibody, you're going to have blood that attacks itself, okay? So this is why you can't mix bloods. You have to be very cautious of what antigens and antibodies are present, because if you put the same antibody in here, it's going to attack the antigen, okay? So you don't want to match those 2 guys. They want to always be opposite. So if I have, in this case, A antigen, I have to have B antibodies floating around out here, okay? Now, these are also known as anti-B antibodies. I just call them Bs, but whatever you want to do is fine. I think your textbook does call them anti-Bs. Here's the reality. I am going to have an antibody that's against B. Okay, so why is it against B? Because I can't mix those bloods. All right, so in this case, you're going to have the opposites going on here. So in this case, my cells for type A have A antigens and B antibodies, okay? We're going to go on, and then I'll come back and do some examples with you. If you are type B, which antigen do you have on the surface of your cell? Remember, my type determines what antigen I have. So these have to be Bs on the surface of this cell. If I have Bs on the surface of my cell, can I have B antibodies? No way, they can't mix. So in this case, I have to have A or anti-A. Okay, antibodies kind of floating around out here. Those, remember, are not attached to the surface of the cell. They're just floating around in the blood. We can move on then and do AB. This one's a little bit more tricky. If I am AB type, that means, remember, my antigen determines what type I have. That means I have to have A's and B's on the surface of my cell. Okay, so this is the kind of the kicker, because if I have A's and B's both on the surface of my cell, can I have antibodies out here? Remember, if I have an A antibody, guess what? It's going to attack my A's. If I have a B antibody, it's going to attack my B's. So guess what? I have no antibodies out here, okay? That makes these guys really special, is that they don't have any antibodies floating around out in their surface. And then lastly, we have type O. And with type O, also, again, very interesting. I tell students to remember type O as being 0. These guys do not have any antigens on their surface. So guess what? If I don't have any antigens on my surface, I'm going to have A and B antibodies floating around out here, okay? And those guys are protecting my cell because I don't have anything attached to those guys. So all in all, what you're getting here is this story of what cell surface marker do I have and what antibody do I have? And to this day, I actually, if I've got a kind of complicated question, I still draw this out, and that's okay. On an exam, I just make this real clear for myself so I can see it. Remember, I don't want things out here in blue to match my orange, because if they do, they're going to attack. Whenever you have the same antibody meeting up with the antigen that matches, so if I were to put these anti-A's in here, those A's would attack A's. What happens there is blood would clump up. That's called agglutination, and that's kind of another little word that we use in anatomy. Agglutination just basically means antibodies attack antigens. Okay, you don't want that. This is not good, all right? But that's what would happen. Okay, so another little thing, let's go ahead and discuss then if I were to mix or what I could do here. So with type A, I have A surface markers and I've got these anti-Bs. Can I put type B blood in here? Okay, if I were to put type B blood, I've got all these surface markers here. And these Bs, would the Bs attack the Bs? Yes, so I can't do that. So the only types of blood that I can get here is A, and in this case, O. You see O here has nothing on the surface of itself. So I can put it in actually any one of these and no antibody can attack because there's no antigen to actually attack. So those are the two blood types I can get there. For B, it's the same kind of story. You can always get yourself. And again, you can get O. Those are what you can receive. I should put that in here. Receive. Okay, when you look at type AB, this one's kind of an interesting one because here, remember, there's no antibodies. So if there's no antibodies, what that means is that this guy can receive anything because these antibodies can't attack anything because there's nothing to attack. So this guy can actually get AB, and O. Now, this guy is known as a universal recipient. Okay, and the reason is because there's no antibodies there. For type O, because he's got nothing on the surface of his cell there, he can give to anyone. We actually call this guy the universal donor. Okay, it can give to anyone because there's no antigens, but guess what? It can only receive from itself, okay? And the reason that is because it's got both antibodies in the surface. So if you give him A, B, guess what? Those both are going to attack. If you give him B, your Bs are going to attack. If you give him A, A's are going to attack. So you can't do that there in that scenario. So once again, this is just kind of a better idea of what kind of blood and how this blood typing would work. The other thing that we'll talk a little bit about in lecture is where the negative comes into play. Many of you guys have heard about positive and negative, and that actually comes from the rhesus monkey, which is where that antigen was first found. But if you are, for example, type A positive, that would mean that you have that Rh antigen also on the surface of your cell. And so it plays the same game. You just have to make sure that you don't have antibodies that are going to attack. This is not a huge ordeal for us because you do not naturally make that Rh antibody. So typically, unless you've had some blood mixing along the way, it's not really that large of a concern, which is why I don't teach it here on this graphic. So we'll talk more about that in lecture, but this is what you'll specifically need to know for getting through lab, and then there will be some questions about this on lecture.